Hello and welcome back. This is the fifth lecture already in the Chapter 12 series. And in the past we've talked about replication, specifically DNA replication and how DNA molecules make more DNA molecules. Then we talked about transcription, which is when the DNA molecule opened up and allowed RNA polymerase to build basically a messenger RNA molecule from the antisense strand. And I didn't talk in much detail about the antisense and sense strand. We may discuss a little bit about that in class, but for now, we're going to go into how that messenger RNA, specifically how that mRNA message will produce a protein. Replication and transcription were both performed in the nucleus, and this is the only process, speaking about translation, it's the only process in that whole central dogma that takes place outside of the nucleus. And so we're out in the cytoplasm, and the mRNA message, which is this long kind of single ribbon of DNA that we can, or of RNA that we can see down here, it will find a ribosome, and we've already talked about the anatomy of a ribosome, but we'll review it. The ribosome is composed of protein and rRNA, and there are two subunits. You've got this large subunit on top, you've got the small subunit on the bottom. This small subunit has a mRNA binding site in it. The large subunit has the tRNA binding sites, and there are three. There is the E site, there is a P site, and there is an A site. Now, one distinction I want to make, when you remember the sites and you look at a ribosome from left to right like we normally read, you might think that it goes EPA, meaning this would be first, second, third, but in reality, even though the mRNA message moves through in this direction, we are going to follow that direction with our tRNAs. And so this would be the first site, this would be the second site, this would be the third site. Okay, and so we're going to work through this ribosome from first to second to third, and move our RNA and our tRNAs in from right to left in order to build the protein. And we'll look at the specifics here in a minute. Okay, three processes or three kind of phases of translation that you need to know is initiation, elongation, and termination. Okay, and initiation is exactly what it sounds like. The mRNA is going to move through this small subunit until this AUG codon is reached. Okay, AUG codon. The AUG codon is the universal start codon. Okay, and so we will always start at AUG. AUG doesn't have to be the very beginning of our mRNA message, but we will always start our translation at AUG. Okay, notice that this message started with maybe a U, C, G, G, U, A, and then we get to AUG. So this mRNA message is going to slide through our small subunit until AUG is found. Once AUG is found, AUG is going to sit, this codon is going to sit at the P location, the P binding site, and is going to wait for methionine. Now, methionine is an amino acid that is linked to a tRNA. The tRNA has an anticodon. This UAC is the anticodon. The anticodon is complementary to the codon. Okay, and so it makes sense. Codon, anticodon, they are complementary pairs of each other. AUG is going to code for UAC. UAC is the anticodon linked to this tRNA message. The tRNA is going to bring our methionine, which is our first amino acid. Okay, notice these little spheres on the kind of top side of our tRNA message or tRNA molecule is the amino acid. Once these bind first at the A site and then slide to the P site, they will stay at the P site until the adjacent or next tRNA message binds to the A site. Okay, now this 3 nucleotide codon down here is going to be the complementary sequence of our anticodon. Again, the anticodon is brought by the tRNA, and the anticodon is actually what allows this tRNA message to bind with our mRNA message. and therefore our mRNA will code for the right protein, and this will be the second amino acid in the sequence. Now, notice right here, okay, this is kind of stage one. This is initiation. Here is the beginning of elongation, and elongation is when we're actually building this polypeptide or protein. This would be our anticodon, this would be our codon, this is our next anticodon, this is our next codon, and you'll notice that it goes in threes. Okay, and so this would be the next codon, next codon. Those codons would bring anticodons, and those anticodons would bring adjacent amino acids. Okay, now this tRNA molecule with the methionine is going to hand off the methionine to the leucine that is bound to this tRNA. Okay, they're going to produce a peptide bond. We've talked about in previous chapters that two amino acids are linked by a peptide bond. Those peptide bonds are actually going to build our polypeptide. When this tRNA loses its methionine, it's going to jump to the E site. This tRNA with the growing polypeptide is going to jump to the P site. Now P stands kind of for protein, that's how I remember it. So that's where the protein is going to be built. A site stands for like acquire or acquisition, right? That's the site that the tRNA molecule is going to bind to. And then E stands for exit. And so as soon as this tRNA molecule loses its methionine, its amino acid, it's going to jump onto the E site, and then eventually it will leave the ribosome as this ribosome overall shifts down the mRNA message that way. Okay, now you can see kind of the next few steps. Once this methionine is handed to this tRNA message and the peptide bond is formed, this tRNA message or tRNA molecule that has lost its amino acid will leave out of the E site. This tRNA will shift to the P site, and then the next amino acid, which is carried by the tRNA message, will bind to the A site. This anticodon, again, is complementary to this codon. Once this tRNA molecule binds to the A site, this group of two amino acids will be linked with a peptide bond to this cysteine, and now we'll have a 3 amino acid peptide, okay, or a 3 amino acid long polypeptide. Once the last tRNA has joined its amino acid to the growing polypeptide, and we reach a stop codon, that's called termination. So elongation has happened, we have elongated our polypeptide, and then a stop codon is reached. There are 3 stop codons. We'll talk about them when you see the codon chart here in a minute. But the stop codon is going to tell the ribosome and mRNA message to stop translation because our protein has been built. Okay. Once the stop codon has been reached, the ribosome is going to disassociate, it's going to break apart. The large subunit, small subunit are going to detach, and the protein that we just built is going to be released by the ribosome and is going to be folded into the quaternary or tertiary structure that it codes for, based on kind of those hydrogen bonds, disulfide bridges, stuff like that. And it will be used where it needs to be used. Okay. So AUG, like I talked about, AUG is the universal start codon. It is methionine. You will always start with AUG. Okay, the next codon in this case is CUU. It codes for leucine. You will not always have leucine as your second amino acid, but notice that you go in threes. So you're always going to code for an amino acid by a group of 3 RNA nucleotides. Okay, so protein. We've talked about it in the past, but just as a refresher, remember proteins are strands of amino acids, and those amino acids have to be together in a specific chain or a specific strand. It is a protein chain built with individual amino acids. Those amino acids are going to be shaped or folded based on hydrophobic, hydrophilic interactions, polar, nonpolar interactions, hydrogen, sulfide bonds, stuff like that. Okay, start codon is always AUG. Stop codon is reached, translation ends. That's what we call termination, and the protein is released by the ribosome. Here is the codon chart. You will have a codon chart to use in assignments and on a test. And you have to know how to use a chart. Some of the charts you get will be circular. Some of the charts will be square like this. And I'm going to go through the square chart because I think they're a little easier to use in my opinion. Notice you have kind of three axes if you think about it. You've got this axis, you've got this axis, and you've got this axis. Notice the first, second, third also. A codon is three. Okay, well this is saying there's three stop codons, but a codon is three RNA bases. And so if our first base is CAU, you're going to go to the first, and the first base is C. So you know that it has to be in this first row here. Then you go second, A. So you go in this row to A, our second base. You know that it has to be in this column. Okay? Then you go to your third base, U. So you go over here. And notice that on this column there's a U, C, A, G. We know that it has to be in this row where all of those intersect. Right here in this shaded region is CAU. That is our codon. That codon codes for histidine specifically. So you'll go first, second, third. Where that first, second, third codon intersects is the individual amino acid that you want. That is how you use this chart. Okay, and so if our codon is AUG, you go first with A, so we're going to be right here. So we know it has to be in this somewhere. U is our second, so here's U, so we know that it has to be in this box. And then G is our third, so we go over here on the third, UCAG, notice right here. AUG is methionine, that is our start codon. You should know that one by heart, okay? AUG, methionine, it is our start codon, that's what we always start with. Notice up here though, there are three stop codons. So you've got this stop codon, UAA, this stop codon, UAG, and this stop codon, UGA. If any of one of those three stop codons are reached, translation stops. We call that termination, and our protein is complete. Okay, that's where we're going to leave it. That is translation. We will talk in our next lecture briefly about specific mutations that can happen and what the total ramifications of those mutations are. Until then,